Welcome back. Welcome back to Covestro Live. Thanks for joining us. I'm Nico Palosua and I'll be your host today. Today we got to talk about biotech. We'll deep dive into white biotech, but we'll see how that stacks up with the other biotech colors. Biotechnology has been used for thousands of years, with early examples including the fermentation of beer or the creation of cheese or yogurt. But with the advancement of science, new horizons have opened up. Also, new demands have risen, the foremost being the need to reduce the dependence on fossil fuels. So, the science of biotechnology may very well provide answers for a bunch of challenges as we transform into a more circular economy. And talking about science, let me bring in two scientists who are really amazing in their vision and expertise on the topic. First up, joining live from Aachen, Germany, we have Professor Dr. Ulrich Schwaneberg from RWTH Aachen and Leibniz Institute für interaktive Materialien. Welcome, Uli. And let me put you guys up. Hi, Uli. Good to see you.
Thank you, Nico. Pleasure to be here.
And from Covestro, another amazing scientist, Dr. Lukas Reisky, or Reisky in German, who heads up enzyme technology at Covestro, live from his Cologne office, I presume. Welcome, gentlemen. Good to see you.
Hi, Nico. Thanks for having me here.
So today we will learn about the different kinds of biotech and the colors associated with them. But before we go there, let me remind you, dear audience, that you can be a part of this conversation by typing in your comments or questions or protests down below in the chat. Here in the studio, we're able to see them and discuss. And just to test your current knowledge on biotech, let me start right away and poll you with a question. And the question is as follows. Who came up with the term biotech for the first time? So just go to slido.com and use the hashtag WBT to participate. Don't Google it. Or as you said today, don't chat GBT it. Fantastic. So let's start with the basics and resolve why we need biotech in the first place. And Uli, this I think comes to you first. Can you share with us what is biotech and why is it so important for the world?
Yeah, no, thank you, Nico. Before starting to go into definitions also on, I think simply put, as you already outlined, biotech is part of our daily life. And I assume that everybody here in the live stream today has already used a product that was made by biotechnological means. So when you ate your bread, you said cheese, you already mentioned, when you do your laundry, when you fill up your car, and there's some bioethanol in there, everywhere in these products, you have biotech. And the importance of modern biotech has recently been in 2018, I think in 2020, awarded with the Nobel Prize for directed evolution methodologies. What you hear in the back is my coffee machine that is shutting down. This is great. And with CRISPR-Cas, I don't think I have to say much. So in essence, biotechnology is a key enabling technology that allows you to do a lot of things, produce a lot of goods that we're using today, and Nobel Prizes you only usually get for something not only for great technology or great methodology, but also if it has an impact on society.
So we know that, and we have heard before that there are different colors of biotech. There's red biotech, there's green biotech, even blue, and even other colors. So can you allude with us a little bit about, as we go into white biotech, what are the different colors of biotech?
Yeah. So personally, I'm not a big fan of this colored application of biotechnology, like red being the medicine, green being plant, or white as industrial biotechnology. So in total, we have 11 colors when you look in the
11, wow.
11 colors. I think that what we really should see is that biotechnology is a key enabling technology. And if you go for many, many applications, and if you go, for example, to Japan and you talk to a person about red or green biotech, they will look at you and not really understand generally what you mean. But I think what is really important and where we hear today, some of the success stories are with white or industrial biotechnology today. And let's see. Mm-hmm.
Thanks, Uli. So now is a fantastic segue to Lukas to talk about white biotech. So Lukas, now being the industry representative, what is white biotech and why are you so interested in it?
Yeah, so white biotech really helps us to produce chemicals in the industry, also for the chemical industry or the polymer industry, and it's already widely applied. So, for example, bioethanol production, production of citric acid, amino acids, and so on. These are common techniques that are used already today, and there is even more research on future processes. So basically in white biotech, we have two areas. First one is, say, fermentation processes where living organisms are cultivated in special vessels. We call them fermenters to produce, for example, a bioethanol. And on the other side, we also have processes that run with single enzymes. So these are single catalyst proteins, and they can also replace some chemical processes that where they have some advantages. In general, white biotech is already applied as said, and we are even looking for more applications and more examples where we can apply it.
And I'm looking really forward to see the examples and also to hear a little bit about what the opportunities are. And Uli, 11 colors. But if we talk about white biotech, what are the new options that you see in this space?
Yeah. Nico, let me start with two circles, since we are talking also then a lot about polymers and applications in plastics as a new application field. So the polymers and plastic are huge bulk products produced with more than 3-digit million ton scales per year. And we use them in all kinds of products when you look around, foils, shoes, and so on, because of their excellent properties like lightweight and so on. First cycle. The second cycle is that we have, how to say, if we have to preserve our natural resources. And if we want to preserve also our living standards in, let's say, climate neutral circular economy, we have to develop technologies where we basically use products, polymers in a multiple way or in a circular way. And second circle. And you ask me, what can biotechnology do? Biotechnology can exactly help to solve a lot of challenges that all the many material streams that we have, that we use them in a more circular and more sustainable way. When you look, for example, at PET, the depolymerization of PET, the activity of enzymes, of catalysts, doing this depolymerization is already reaching productivities like the cellulases doing with cellulose for bioethanol production. So when you put this together, then also for many other polymers, you see the potential that biotechnology offers there.
I think that's fascinating because many of us actually are in touch with biotech products without even knowing it, and this is something that we'll also ask our audience going forward. Lukas, let me come back to you. You mentioned microorganisms and enzymes. How do you exactly use them? I mean, how can you use them in chemical processes?
Yeah, we use them in special vessels, reactors, fermenters. and they can really help us to simplify multi-step reactions. So reactions that by chemistry would take several steps and purification steps, they could potentially be run in a single cell in one step, basically. Take as an example the conversion of sugar, glucose to aniline. That is also what we are doing at Covestro. That there are many steps in between, and probably that can only be done by biotechnology. And in that way, biotech can even open up new options for products that are not reachable with classical chemical methods. So another advantage is that we can operate under very mild conditions, because the catalysts they are very selective and specific. Together with the mild conditions, we also don't produce many side products. Usually at the temperatures and pH values, there are not many spontaneous reactions. So we can also generate a very pure product without side streams, and also avoid, in many cases, toxic solvents that would be there in the chemical counterparts. So all in all, this is how we use it. We can also use it to incorporate renewable resources, as said, these sugar materials. And I think it has a great potential to also revolutionize the industry in future.
And again, it's fascinating. It's fascinating what you can do with biotech, going into areas that many people, frankly, probably don't even know about yet. Let's see what our audience has come up with regarding the first question. Let me just go there and take a look and see what we have. Okay. Uli, what do you think about the results?
It shows that the majority of the business are knowledgeable.
So the right answer, who coined the term biotechnology? Lukas, can you talk a little bit about the history?
I just remember from lectures that it was an Hungarian name, so otherwise I wouldn't be able to answer these questions.
Yeah.
Of course, I remember Louis Pasteur. Faraday, okay, is out of the field. Pasteur also did a lot with microbiology in the 19th century, I guess. But I think it was Karoli Erecki who termed who named it biotechnology in the end.
Yeah. And that's the right answer, and it really, I think, shows the history. This is not something that came up yesterday. People have been working on this topic for quite a long time, and now we're seeing that huge acceleration on the topic. So Karoli Erecki is the right answer. Let me go right away to the next audience question.
Maybe a comment on that, Nico. I mean, he termed it biotechnology, but humankind was using that thousands of years before in producing wine and these different vinegar, etc. Using it, but without calling it biotechnology. So it's with our culture for a very, very long time.
Absolutely. And I have to say, I'm a big fan of those biotech engineers who really know how to make good foodstuff products. I think that takes a lot of skill. And especially in Germany, you have quite skilled people who specifically in the beer section. Good. The next audience question is up. What biotech products have you personally used so far? So again, go ahead to slido.com with hashtag WBT and pitch in. Now, let's go to circular economy and talk about a little bit about the role of plastics in the circular economy. Because we all know that we are trying to avoid single-use plastics as much as we can, like with the bags that you cannot get in many countries anymore, and they're even banned. But on the other hand, we need even more plastics to materialize the sustainable living, something Uli you also just alluded to in earlier. So as the demand for plastics will increase in the future, because we just cannot do without them, the industry really needs to come up with innovative and more sustainable solutions. So let's talk a little bit about how biotech exactly can support circular economy. And maybe, Lukas, we start with you. What's your take on what is the role of biotech in supporting the circular economy?
Mm. Yeah, maybe first of all, for the circular economy to realize that, we need different, say, pillars or areas. Yeah, first of all, we have the linear value chain from crude oil to the polymers, for example, at Covestro, polyurethanes, polycarbonates in the use cycle. And then at the end, they are thrown away, burned, landfilled, whatsoever. Not very sustainable. Yeah. And we need to change that. So what we want to do in the first place is to use these old materials, the waste, and use that as a feedstock to produce new plastics. Yeah, that is basically recycling, keeping that carbon in the loop, and by that also getting rid or reducing the use of crude oil. Yeah, this is also an important aspect. And when we have to use new carbon, we would like to use then alternative resources. That could be bio-based resources, plant-based, as such, sugar, starch, but could also be, for example, CO2 and or syngas, for example, to fuel that cycle. And finally, in the end, to really run all these plants, etc., you also need green energy, of course. Otherwise, you will be producing the CO2 just in another place. And for two of these aspects, I think biotechnology can contribute. So first one is obviously renewable resources, plant-based materials, sugars, etc. When we have a fermentation production, for example, with the bioaniline, where a very important intermediate for the chemical industry is produced based on plant biomass and not anymore on crude oil. That can really help. And the second one is recycling. So where we are also working on a thing called enzymatic recycling. So we are trying to break down the polymers into the monomers, so the building blocks, by the use of enzymes. And I also brought a small sample here. Let's see if the camera can focus that. So what you see here is a piece of foam, PU foam, in an aqueous buffer. Yeah. This is as good as it gets. And on the other side, this is what happens when you add some enzyme to it. So there was the same piece of foam in that tube, and it's completely gone. Yeah, completely dissolved into smaller building blocks, smaller particles. And now the aim is to really isolate them from these solutions and rebuild that piece of foam. So these are the aspects where I think that biotech can contribute to circular economy.
That's fascinating. I mean, the foam is being eaten up and transsolved into other products. That is truly amazing. I don't know, Lukas. I know we have a 3D model that we can actually deep dive into a protein and into enzymes. Do you want to carry us through a little bit about the science behind that?
Yeah, maybe I can make a start here. So that is also when you show the model. So it's a 3D model of a protein, basically. So our little helpers, our catalysts. And we also want to engineer those, so meaning exchanging amino acids. But here I think I can hand over to Ulrich because he's the real expert for enzyme engineering and directed evolution.
Thank you, Lukas. So the molecule that you see there in a protein, they have usually, let's say, sizes from 3 to 6 nanometers. So when you think about the thickness of your hair, you make it more than 1 million times smaller, then you slowly reach the size of these nanomachines. And what basically nature does, it's really a very well 3D defined molecule, and at every position, I think now you zoom in, you see this sticky side chains there. Nature offers basically to put in specifically 20 different building blocks in one position. And when you imagine that such a protein has hundreds of such amino acids, if you would only compose out of 6, so in one position 20, two positions you have 400, 20 multiplied with 20, 8,000, 160,000, 3.2 million, 64 million different variants only with 6 of these positions. This basically gives you a feeling how powerful these enzymes are, and why you can make antibodies so specific, and why you can engineer them in a way that they can, be tailored to cost-effective production conditions, and then really push the conversion. So it's really beautiful. So technology-wise, generating really millions of variants in 2 to 3 hours. This was solved already in the 80s by Loeb. And nowadays, you have basically screening and selection technologies to really sample through these millions of variants to find the ones that are more stable, more active, are not affected by molar concentrations of substrate or product.
This is it's so high tech. It's crazy to think about the level of technology that you're employing to get to the bottom of the opportunities of biotech. And Lukas, let's talk a little bit about your unit. The Junior Research Group Enzyme Catalysis, or NENSI for short. And I don't know how you get to the acronym NENSI from that, but maybe you can share us what's the unit and what's so special about it.
Yeah, first of all, the acronym, a colleague came up with that nice idea, so we took it then. What is so special about it? So the unit is there since summer of last year, and it's also publicly funded by the Ministry for Education and Research in Germany. And the special fact is that we are establishing a group that is located in 2 different areas, I would say. Our part is at Covestro, connected to the Competence Center of Biotechnology, and another part is in Ulrich Institute at the RWTH in Aachen in academia. So we could call ourselves a bridging group, and that is, say, something novel. I've not seen that before, but in my opinion, it makes a lot of sense, because our aim is really starting from basic research, early research, but with a focus on the application. And early research, protein applications, enzyme engineering, that can all be done in Aachen, and Covestro can then contribute knowledge on upscaling, life cycle analysis, also evaluation of the concepts that we might develop, a lot of knowledge on these polymers and customers, so that we really can make the connection from the early research to the actually realization in industry. And that is the aim, and also I think that the unique thing that we are having with that group. Ulrich, do you have to add something on this?
I think it's really a very nice description in the sense. So what I love about it is that basically there is also a long-term commitment from the industry side, which we have very seldom. And I think we need more of these models and also to transfer certain technologies and to interdisciplinary training than already PhD fellows exactly at the interface. So I think it's really great.
And it's great to hear, Uli, your commentary on, I mean, this is a public-private partnership that I think extracts more value from the collaboration as compared if academia and the private companies would work on their own. Mm-hmm. And maybe we can, talk about the other collaborations that Covestro has already done. I mean, we have some collaborations with Covestro in the past, research and development corporation, other in biotech, which have also been involving private entities. So, Lukas, can you maybe talk about that history?
There are some nice examples. I mean, Covestro as a company is relatively young, 2015, carved out from Bayer. There are quite a few examples. For example, the bioaniline project, which I mentioned, and that is one that I can also mention here publicly. Where we are working together with different partners, so be it companies, but also a lot of academic partners, to really make it possible to produce aniline, which is a central part for polyurethane production, now also from bio-based feedstocks, and that is a breakthrough actually. There are other examples, for example, collaboration with the US company Genomatica, or Geno as they call them now. And here the aim is also with the fermentation process to produce another diamine, HTA, that can be used in coating and adhesive applications. And that was also produced previously, or is still produced, based on crude oil. These are 2 areas, and hopefully the Nancy Group will also be a success story in the future. So we are only here for half a year right now, but I'm very much looking forward to the next 5 years where we are funded, and hopefully we can also present some great results in the end.
And I'll ask you about the expectations, also Uli from you soon. Before we go there, let's take a look a little bit about what our audience has said about their use of biotech products so far. Let's see what we have here. Cheese, wine, vaccines, and beer. how do you evaluate this combination of biotech products, Uli and Lukas?
Sounds good. I guess the people are coming more from the east where it's already later. Otherwise, I mean, depends. But yeah, right. These are all biotech products.
Mm-hmm. I agree. And important ones.
Yeah. So certainly there is a lot still that can be done to let the people know about where biotech can positively influence the lives of us. But I'm happy to see that vaccines was there. And actually we could see BioNTech was in our audience. So thanks BioNTech for doing your biotech research on the COVID vaccines. I think everybody is very appreciative of that.
Mm-hmm.
So, and as I said, let's talk a little bit about where this is going, what's coming next. And maybe we start, Uli, with you. What are your expectations where this biotechnology, and specifically maybe white biotech, is able to get us in the coming years?
I think one strength that we have here in Germany is that we have many bigger companies that are really very, very active in R&D. And this is really something that many other countries do not have. And if you ask me where to go in the future, what I would perhaps more wish for is that we here in Germany really push to become a leader in the biological transformation of industries. This is a big word, biological transformation of industries based on renewable resources. But the exciting part is that now we have basically the push regulatorily, we have the push from the resources. We know which resources get more and more available. And my wish would be that we do not stop by making existing products from renewable resources. But that for us, this might sound trivial, but that we combine it with developing technologies, processes to couple it with a climate neutral and a circular economy as a first step, so that we can preserve our living conditions and nature, but then also continue from there further to integrate then also, for example, biological functionalities in and on materials to introduce a programmable degradability. If you have 1 kilogram of cotton and you put an antimicrobial coating, you can make curtains that you can perhaps use in the hospital. If you put a water repellent coating, then you are with clothes for outdoor, which has then a completely different value in it than the biomass itself. And hopefully we are then smart enough in the sense like Covestro is basically thinking about in a circular economy, that we also invest in key enabling technologies. So for example, if you have switchable glues and you design products already made for recycling with switchable glues with different triggers, then you can have a completely different speed in a circular economy. And I hope that with protein engineering, we can basically also build triggers in proteins that they can become programmable and that we push in that direction.
When you only talk about this, it doesn't sound like science fiction anymore. It sounds like actually reality is coming in a much more circular way, which I think is very, very impressive. And we actually thank you for all the questions we are getting from our audience. We'll get to them soon. Before we go to the questions, Lukas, what are your expectations for the years to come? Accept the high, expectations from Uli, of course.
I think there was already quite a good outlook by Ulrich. I think that the field will, at least in my area, enzyme technology, will speed up even more. So we will see that gene synthesis prices will fall. Computer programs are really speeding up the enzyme engineering. Also with AI in that area, there was a lot of progress in the last time. And I think that will continue and lead to really that we can speed up these developments and not take, say, months or years, but only then weeks or months until we reach the goal. And yeah, in industry, time is money. That will definitely help us.
Thank you. We're really looking forward to kind of that future. Let's go to the questions. Let's see what we have. We have some, I think, really good questions from the audience. We have Mr. Raju asking, how do we see bioreaction versus catalytic reaction with respect to energy balance, especially when bioreactions happens at milder conditions? Who wants to take?
Yeah, I can perhaps start here. So I'm always not a big fan of saying, oh, one is better than the other. So I would always look for something where it's more beneficial. So one advantage that I see, especially from the enzyme side, when you do recycling of at least polymers that are connected through Eastern or similar bonds, is that with enzymes you can basically material in a specific start a degradation. So you should get much more defined product mixtures, which perhaps then open up again better or new opportunities in terms of recycling. One main challenge there is always that when you look at nature, how did nature make enzymes that are very efficient, for example, integrating cellulose. There they have a specific binding domain that binds there and then degrades like a Pac-Man the cellulose. When we now go to man-made polymers, many of these enzymes, they don't have, let's say, this specificity or this binding domains to do so. So basically what I want to say is, on the one hand, I see the advantages on the product side, product composition, perhaps also in terms of energy. But we have then still to do a lot of development for the depolymerization, and then there will be simply polymers where very likely chemical treatments will be much better. So when you think about polymers that are purely made out of carbon, so when you have to use ozone or metal catalyst or peroxides or whatever to break the CC bonds, this is something that are not conditions that enzymes can tolerate very, very well. So at the end, it will be again very likely a combination based on the composites of your materials, because most products that we at the end have to recycle, so even foil and so on, it's not a defined material like a PET bottle. It's basically made out of many different composites. And I know that is a very general answer, perhaps more from the academic side, and I would give over to Lukas what you think.
Well, on that specific question, regarding the fact that biotech reactions in most cases need lower temperatures to run, I think there is an advantage on energy cost or the amount of energy that you need to put into these reactions. Yeah, when we compare it to a classical chemical reaction, 100, 150, 200 degrees centigrade, of course there will be advantage here.
Excellent. Thank you so much for the question, and thank you also for the great answer. We have another question, I think, going more into enzymes. Let's take that question. Is from Haliang. Thank you, Haliang, for the question. The question is as follows: what's the advantage of using enzyme only or using strain directly for degrading plastic? You want to go first, Lukas?
I can go. Yeah, so when we use only the enzyme, that is a single catalyst. So that will cleave the plastic and leave the building blocks in the solution as they are. Now, there are also other approaches where you use whole cells that produce the enzymes themselves, for example. And in these cases, these cells take up the monomers and eat that. Basically, they use that as food. Yeah, then they transform that into biomass, maybe other products, but then these are not the building blocks anymore. So we prefer the first option, pure enzyme, pure building blocks that we can isolate and put back to the polymer. No organisms that will eat up our precious building blocks.
Thank you, Lukas. Do you want to add anything, Uli?
I think you are on
One
mute.
thought that I can perhaps add is when you want to do recycling and when you collect the plastics and so on, with enzymes, you don't have to be too much, how to say, worry about that there are other contaminations in there biologically from molecules that might even be harmful to strains or other spores and microorganisms. So from this point of view, I would expect that with enzymes you can make the more robust or reproducible processes.
Thank you very much. We have researchers out there who already want to start working with enzymes. So let's take a question from Murali Reddy from LinkedIn. Is this enzyme for PU available for R&D projects?
Yeah, we have quite a bunch of proprietary enzymes, but Murali, if you want, you can write me email or contact me via LinkedIn, and then we can discuss it in more detail.
Perfect. Thank you, Lukas. And it's great to see that interest in research and science. There are many people, I guess, who want to, discover the benefits. Let's see. And actually, there's another question, I think, going in the same direction. Let's see about that. Bravo, Covestro! Bioenzymatic reaction to degrade plastics or replace other catalysts are welcome in our world. Are there public papers on these topics? From Bernie.
Ulrich, do you want to go or should
No,
I?
go ahead. It was Bravo de Covestro. So
Okay.
talk about your papers.
Yeah, there are papers, mostly on polyester degradation, or polyester recycling with enzymes. That is the biggest topic. Now also more and more coming from for polyurethanes, and we ourselves with another collaboration partner just recently, I think a week ago, published a new paper on enzymatic polyurethane degradation and recycling. So there are papers, and by typing into Google Scholar, you will be able to find them.
Thanks, Lukas. Uli, you want to add?
No, nothing. That is fine. Mm-hmm.
Cool. Great. So I think we have covered most of the questions, and we are actually at the end of our broadcast time. So thank you very, very much, everybody, for joining in with your questions and joining our Slido questions, where we kind of discover a little bit about where biotech is coming from. Thank you, Ulrich. Thank you, Lukas, very, very much for joining our live talk today. And thanks to our wonderful audience for joining with your enthusiastic and sometimes detailed questions and comments. Stay tuned for more exciting lives from Covestro, and have a wonderful day. See you guys.
Thank you. Bye.